Testing, testing, 1, 2, 3. Alright, I got no clue what I'm really doing, but let's get right into it. So, I am currently taking a master's for synthetic biology over in the United Kingdom, and I wanted to start putting out something on a channel where I would basically just try and put together different sorts of, plasmids or DNA sequences, whatever, for all sorts of fantastical things, and just for practice and try and get some feedback both on the quality of my work, spot things that I maybe missed, as well as suggestions for new things for me to try. But before I can do that, I figured it might be a good idea to kind of give like a brief overview of like the very basics of kind of what I'm working with here. So, and by what I'm working with here, I kind of mean synthetic biology in general. And basically, it's going to be like, designing whole little strings of DNA, from one end to the other, and you put them inside a big cell, and then, tells the cell to do things, whether it be bacteria, archaea, yeast, what have you, though most of them are probably going to be large multicellular organisms such as, well, for example, a black lotus plant, or say a lemon that bursts into flame. Yes, that's right, Cave Johnson lemon. But anyway, as far as like the basics of synthetic biology. So what is synthetic biology? Basically, it's what Hollywood thinks genetic engineering is. Genetic engineering, in its most part, is just taking a gene from one organism and putting it into another. Synthetic biology, on the other hand, is actually taking a gene or different parts of a gene and using them to craft your own gene, basically creating an entirely new gene from scratch. Or I say from scratch. But at its most basic, a gene can be divided up into 4 different parts. So let's see if I can break it down real quick so it's nice and simple. You have the promoter. Promoter is basically the segment that says, hey, here's like when to make the thing. For the most part, that's one of the key things that a synthetic biologist cares about. Then you have a ribosome binding site, usually using this symbology, just like a little half sun thing here. And that is basically where the enzyme binds to the DNA strand, like where it physically attaches to start reading through the thing. Then you have the coding region. Now, the coding region can be lots of things. There's like introns and exons and all that stuff. You don't got to worry about that. All you got to really care about is the coding region is the thing that's made. This is like the spider silk or the glowing protein or the what have you. Like this is the part that like the coding region is the part that is what the gene does for the most part. So the two, and then finally, last but not least, you have the terminator, which is basically just a segment that says, okay, stop, cut off, we're done here, that's the end of the gene. It's like a period at the end of a sentence. So that's pretty much the most basic part, or that's pretty much like the most basic kind of aspects of synthetic biology. What you're doing in synthetic biology is you are taking these different parts from all sorts of genes, from all sorts of different organisms, and you are rearranging them. I kind of don't want to say like Lego, but yeah, kind of like Lego. or more like words, really, to create your own sentences. And you can combine these and many other sentences that you've put together to create whole new concepts, maybe new paragraphs, and that whole thing. That's basically the gist of synthetic biology. Now, the RBS, the ribosome binding site, is pretty much, yeah, pardon my terrible drawing here. I am drawing with a mouse. But yeah, this part is basically the same, just about just about as long as it's within like the same organism, and even then, like across a bunch of different organisms, it's not going to change too much. I mean, there's going to be some types of organisms depending on how far apart they are on the tree of life. But like, say, I don't know, a mouse and a chimp, just off the top of my head, it's not going to be that different. If it is different at all. I don't know. Terminator, that's like a physical thing. Like, it winds up actually forming a little, the DNA actually winds up going along and then forming like a hairpin turn. Yeah, form like a little hairpin turn thing, like a little loop. It just forms like a big circle at the end, and that physically acts to stop the enzyme from going any further. It's like a physical thing. It stops it so it can't actually, proceed. So terminators generally are going to be the same anywhere you look. There might be some that are slightly better than others, maybe, but yeah, they're pretty much going to be the same across all organisms. Really, the key thing that you really got to worry about, and what really kind of like the terminator and the ribosome binding site are kind of like the punctuation in a sentence, to use that metaphor some more. The key things, the two key aspects are when to make the thing and the thing to make. Those are the two key aspects in synthetic biology that you really got to worry about. So if I wanted to, say, make a lotus plant that had dark purple leaves, or dark purple petals, let's say. So it looks like that black lotus on a Magic the Gathering card. Well, I would take a gene, I would take a coding region, sometimes abbreviated CDS, coding segment, whatever, and that would make, say, a dark purple protein, like for coloration. And you can find lots of different ones for all sorts of different colors that you can imagine. Pretty much every color of the rainbow. Just because it's so easy to see, yes or no, the thing got made. So it kind of gets used as like a testing, sort of like a testing thing for a lot of genetic engineering or synthetic biology, just verifying that, yep, the thing got made. And then you would attach it along with the appropriate ribosome binding site for, say, the lotus plant, and then a terminator, to a promoter that was specifically only expressed in, say, the flower petals, or the areas that you would want the coloration to be expressed. So you would find, say, a particular other gene that was only expressed in those areas, and then use that gene's promoter so that whenever that other gene is turned on, your gene is also turned on. That would be one trick. There's all sorts of other little avenues and things like that you can play, and sometimes there's weird little like tags that get stuck onto things here and there to say take a take a product and then export it somewhere, or do this with it or do that with it and whatnot. But that, for the most part, like the vast majority of the cases, you just got to worry about promoter, ribosome binding site, coding region, and terminator. And of that, you really only got to worry about promoter and coding region, when to make the thing, and the thing that gets made. And just using those two things with the right, mixture of other stuff, you can do all sorts of crazy things. And that's what I love about synthetic biology is we have an idea of, what is possible in a computer program. Like, we have at least developed some sense of what a program can and cannot do, at least generally speaking. But we don't yet have that kind of internal sense when it comes to biology. And I love that. I love just the sheer craziness where we can't even imagine the things that are coming down the pipeline. If you were to compare synthetic biology to a computer, we are in the 50s or 60s right now. Keeping that analogy in mind, picture the internet. Picture a smartphone. That's the sort of stuff that we have coming down the line. And this is the coding language that it's going to be written in. When to make the thing, and the thing to make. And the neat thing is you can have the thing to make affect other promoters, telling them when to go ahead and make their thing. So you can have one gene split into multiple and do all sorts of other crazy stuff. Or say have coding region only work with something else, like some little other product that came in, and change, and then it activates a promoter. Or it has to say it has to combine with some other coding region, and you need two of them. And when you have two of them together, then it actually binds and activates the promoter. And there's all sorts of crazy little things you can do. But this is pretty much the basics of it. Is like promoter, coding region, along with ribosome binding site and terminator, but the big ones are promoter and coding region. And so just using these kind of basic tools, I'm going to try and create a couple different plasmids or DNA sequences and see if I can explain why I made them the way I made them, or why I designed them the way I designed them. I haven't actually put them in anything. I don't have a lab yet. But I'd like to go through and explain why I designed them the way I designed them, and what, in theory, each of them is supposed to do, and why I believe they are supposed to do that. Now, honestly, this isn't really for educating others so much or any of that stuff. It's pretty much just so I can get some practice in, because I need to freaking practice getting kind of cooped up in here. But also just the fact that I'm a master's student and I feel a little bit behind on some of my stuff. And I just really want to grasp the subject. And you can't do that so much on your own without any feedback. And so that's kind of my goal for this whole series, is just to try and get some feedback and some suggestions about other crazy, possible things you could see me do. I know one guy is making freaking spider beer, like yeast cells that produce spider silk. That's freaking awesome, and most people wouldn't even think of that as being possible. I love that sort of thing about synthetic biology. And so, hope you'll pardon all these little doodles and stuff, but I just figured it'd be kind of helpful to give a rough overview of, very rough, of the most basic basics of all the different pieces that I'm gonna kind of be working with when I start, just putting this together. So, yeah, that's all I got.